Hello everyone, my name is Adrienne Campbell and I am a student at University of Maryland Global Campus. This is Biology 101. This is my PowerPoint presentation of bioprinting and its future in medicine. First, let's take a look at the history of bioprinting to better understand the bioprinting we have today. Bioprinting started in tissue engineering. Then, with the emergence of 3D printing in the 1980s, this laid the foundation of bioprinting we have today. It's used today in mainly clinical trials on the tissues and organs. While it is mainly small organs, researchers and scientists are taking these small steps to achieve their future goals in manufacturing fully functional organs for organ transplant patients. There are three primary types of printers used in bioprinting: inkjet bioprinting, laser-assisted bioprinting, and extrusion-based bioprinting. Each printer has a specific strength. Inkjet functions similarly to traditional inkjet printers, but unlike the traditional inkjet, it is filled with biological ink, comprised of cells, hormones, and building blocks to build a tissue and an organ. Inkjet bioprinters are able to create fine, high-resolution structures, but this printer struggles with printing thick, complex tissue. Laser-assisted bioprinters use lasers to position cells perfectly. The lasers focus energy to specific points, making the bio-ink to fuse together. These printers are highly acute and are used for making micro-scale structures. In the future, it could also be used in tissue engineering. The last type of printer is extrusion-based bioprinting. This bioprinter works by the bio-ink solution passing through a nozzle for a controlled stream. Extrusion-based bioprinting is best for creating thicker, more complex tissue. Each bioprinter has different strengths and weaknesses. The goal determines what printer is used, but the extrusion-based bioprinter is used the most out of all three of these types. Here are some benefits of bioprinting: tissue replacement, organ replacement, drug testing, research advancement, animal testing reduced. Tissue replacement. Bioprinting offers the potential to create functional tissues for medical procedures. This is particularly beneficial for patients in need of tissue transplants due to injuries or disease. Bioprinted tissue can closely mimic the patient's own tissue, reducing the risk of rejection and the need of immune system depressants. Organ replacement. Patients in need of life-saving organ transplants could receive bioprinted organs that match their specific requirements, DNA, which would reduce the wait time and save lives. Bioprinting tissues and organism models can serve as a valuable tool for drug testing and development. Pharmaceutical companies can use these models to screen potential drugs more accurately, leading to the discovery of faster and more effective medications. The research advancements that bioprinting technology has in a variety of fields includes tissue engineering, biomaterials, and 3D printing. Researchers can use bioprinted tissues to study disease and gain insights into complex biological processes without harming individuals and, in the next point, animals. Having bioprinting has the potential to significantly reduce the need for animal testing in pharmaceutical and cosmetic industries. By using bioprinting and creating organs, scientists can directly test the infected organ rather than a whole animal. These benefits will transform the healthcare environment and make scientific research easier to conduct and more ethical and peaceful. While there are positive benefits to bioprinting, there are a few drawbacks and challenges that will continue to impact the evolution of this field of study. Tissue complexity, functionality, ethical considerations, cost, and time consuming. All of animal cells and tissues are complex, and replicating the complex functionality of human tissues and organisms is significantly challenging in bioprinting. Achieving the necessary cellular organization and tissue-specific functions remains difficult for many tissues. Functionalities. Ensuring that bioprinted tissues and organs function as intended is a challenge. Bioprinted structures must not only look like the natural tissue but also perform their role. This becomes more difficult with the more complex organs, which have specific functions to clear the blood or remove toxins. Ethical considerations. The ethical impact of bioprinting is a complex issue. Questions about the source of cells and potential misuse of the technology needs careful consideration. Ethical guidelines and regulations are essential to ensure responsible and ethical use of bioprinting technology. This can provide that line in the sand that separates the benefits that science has from the things that should be classified under abuse or malpractice. Cost. Bioprinting technology is expensive. It involves specified equipment, material, and skillful personnel. The high costs associated with the research and development, as well as the production of bioprinted tissues and organisms, until the price of these expenses can go down, bioprinting will not be entering into the mainstream of treatment options. Time consuming. Bioprinting is a time-consuming process. Creating complex tissues or organisms can take weeks or even months, which may not be feasible for patients in urgent need of transplants. These challenges address the ongoing efforts to make bioprinting more accessible and more medically available. Researchers need to work together to overcome these obstacles to better treat patients and others in need. The future potential of bioprinting has a lot of promise, and it is poised to revolutionize various aspects of healthcare, biotechnology, and beyond. It will impact education and training, organ farming, human and AI hybrids, and longevity solutions. Bioprinting technology can enhance medical education and training. In the future, aspiring healthcare professionals may have access to realistic bioprinted tissue models for practicing surgical procedures, leading to important surgical skills. This would give them the experience they need once they leave school and join a hospital or healthcare practice. The concept of organ farming could become a reality in the future, potentially growing organs on a large scale and addressing the global shortage of donor organs. This could significantly impact the accessibility of life-saving organ transplants. The integration of bioprinting with artificial intelligence, AI, could lead to the development of human-AI hybrids beginning. These hybrids might combine bioprinted tissue and organs with advanced AI technology, enhancing cohesive abilities. This has always been something found in science fiction, space adventure novels, but with bioprinting and the incorporations of AI in biology, it is only a matter of time before these things become a form of reality. Bioprinting might play a pivotal role in expanding human lifespan. In the future, individuals may receive bioprinted organs replacements as a routine part of health care, especially combining age-related organ illnesses. The ability to replace aging or damaged organs with bioprinting versions could significantly contribute to increased longevity and overall health. These future potentials of bioprinting demonstrate the transformative nature of the technology. As bioprinting continues to advance, it holds the promise of not only improving healthcare, but also reshaping aspects of education, technology, and human longevity solutions. However, ethical considerations and regulatory oversight will be critical in guarding the responsibility, development, and application of these innovations. Thank you for listening to my presentation. If there are any questions, please reach out to me. I am available through UMGC. All the sources I use to gather this presentation are in the last 3 slides of this video. I hope you could learn something from this school assignment, and I look forward to doing a similar project like this in the future. Thank you.